last week, and obviously we don't have a lot of time. This isn't a substitute, as you know, as I mentioned last night on the announcements. It's not actually a substitute for actually going through the lecture slides beforehand, listening to the online lectures, if you find that helps. The online lectures, meaning the recorded ones, and doing your readings. You should have done all of that before you actually arrive here. We're not going to go through every slide. I'm going to skip through, and I've decided what the best way to do these sessions, because, as I mentioned, arrived at Swinburne last year. They do things a bit differently here. We do the recorded lectures, but then we do these online ones when there's just too much information. What I want to do, I want to go through and cover the stuff that you're actually going to be more examined on, that I can guarantee that you really need to know, and all the other stuff is really to enhance your understanding of everything. How does everyone feel about that? Yeah, fantastic. All right, great. Because I'd rather it be you take something, a couple of points that are really helpful away from this, rather than me just quickly go through everything that you've already gone through. Okay, and you'll have to excuse my voice and my nose. I'm just getting over a cold at the moment. I spent my entire long weekend just feeling absolutely like struck down with man flu, pretty much. So, all right, so let's have a look at these. So what do you need to know from the chemistry part? You don't need to know a lot of this for the actual exam. It's really about creating a good foundation for you to be able to appreciate anatomy and physiology at a gross level. And now what I can tell you that you'll need to know about is you need to know the basic structures of, essentially, you should really be already know anyway, what the valence shell is, and what the difference between a molecule and a compound, things like that. We'll cover really the main things I want to focus on. This is water, different bonds, and pH. That's pretty much the main things that I'm going to cover in this, and then we'll go on to the next one. So let's skip through some of these. So now you should know this, right? The basic structure of an atom is you got your nucleus, you've got a proton in the middle. So your nucleus and your proton, your protons and your neutrons basically hang around in the nucleus, which is the mid part of the actual of the atom that you can see here. And on the outside, we've got our actual within the electron cloud. They call it an electron cloud because basically what happens is, obviously, as you guys know, there's no actual rings here. They're imaginary, right? That's just gravitational sort of attraction there that you've got. So, but they hover around, and you get 2 on the outside, you get 2 in the first shell, and then you'll be able to have 8 on the following one. Now, the rules do change a little bit as we go on. Do understand that oxygen is the most dominant element that we're comprised of. However, when you actually dehydrate a human, yeah, I know that sounds a bit funny. It's not like a powdered sort of human in a can or anything. But if you dehydrate a human, we're largely carbon based. So carbon is the most dominant element that we have. However, given when we're in our mobile state and we're alive, we only make up around 18.6% of carbon because we're made up largely of oxygen. Once you remove all that gas, then I think that distribution goes up to something like 55% or something carbon. So we are largely carbon-based organisms. Obviously, there's a whole bunch of other things that are relative, that are required for life. So this is what I was saying here. So if you take lithium, atomic number 3, that means it's going to have 3 protons, 3 neutrons, 3 electrons. And obviously the first 2 make up the valence shell. The third one goes over here. That's pretty much all you need to know in regards to the basic structure of that. You need to know that a molecule is 2 or more atoms joined by strong bonds. So you can have two hydrogens joined together, H2. That's a molecule of hydrogen. Whereas a compound is 2 or more different atoms joined together by strong or weak bonds. Water is H2O. So take that molecule of 2 hydrogens, add an oxygen, and you have a compound. And the molecular weight is basically the sum of all the atomic weights. Don't get bamboozled behind mass number. Forget about that. Atomic number is all you need to know. Don't forget about it, but just don't get it confused. Okay. The basic bonds associated with life. Ionic bonds are basically between charged species. Species meaning when we talk about atomic species, meaning different elements, right? So in this case here, this is the formation of salt. You've got sodium. Basically, it's got a lone electron over here. Chlorine basically has a empty space. So what it does is it donates, sodium donates one of its electrons. Remember, it's balanced over here. Now, a lot of people get confused. They think, okay, this means that in its neutral state, this is negatively charged, right? You go, no, not really, because it's balanced because if you've got, look here, there's two electrons here, and then you've got, 2, 4, 6, 8, and then another one over here. If you add up all those electrons, sodium is going to have the same amount of protons and neutrons over here. So in this case here, it's stable. When it becomes positively charged is when it donates one of those electrons to fill up the chlorine outer valence shell, loses that outer shell. It's now got a positive charge. Chlorine has got an extra electron relative to everything else, but that's got a negative charge. Now that's got a negative charge. Then you basically have it so that there can be a opposite attract sort of scenario going on as well. And that's basically the ionic bond. Everyone else can see it? Yeah, great. So if you have problems with it displaying, maybe just exit out and come back in. People have found that in the past. Okay, covalent bonds are basically where you have sharing of bonds going on as well. Covalent bonds are really important for those of you who are going to go off and learn biochemistry and doing a biomedical degree or something like that. And that's because basically it forms all of the really strong structural sort of bonds that we have in our body. They take ATP to break. Ionic bonds don't, and also hydrogen bonds don't as well. So these covalent bonds are strong, and it's where they share the electrons. So they're basically sharing a valence shell. They're connected in that sort of sense as well. That's why it actually takes ATP to be able to break those bonds. Oh, one little thing, just so you don't get bamboozled, we rarely ever get triple bonds going on in biology. So not a lot of triple bonds going on, mostly single and double. Okay, hydrogen bonding you need to know the basics of, and basically that is where you have basically a charge going on between from a hydrogen group. So you can see here, these are really important if those of you who are interested in DNA or proteins. These actually are what hold the double helical structure of DNA. And these are really easy to break, easy to come back together as well. They don't take any energy. If you think about DNA itself, there's a lot going on in the nucleus. DNA unwinds a lot. Has to kind of disassociate from its proteins that it's wound around to be able to allow genes to be transcribed and stuff. And then it has to go back into that double helical shape. And this basically allows that to happen. You don't need to know you don't really need to know a lot about this sort of stuff. You just need to appreciate the very basics of them as well. Okay, so I'm going to skip past all of this stuff here. Oh, one little odd fact here. A condensation reaction, if anyone's listened to my online lectures, how much water does an adult human usually produce through condensation reactions internally every day? How much water do we get? We're meant to drink, 3 liters a day or something like that of liquid all up? Yeah, exactly, half a liter. That's right. Nitya got it right. So condensation reactions actually produce, so this is the thing, if you're not doing anything, you're not drinking anything, you're not eating anything, you're just sitting down, chilling, you are basically producing around half a liter of water a day through condensation reactions. And that's because every time you split these up, these particular, what we call dehydration reactions, usually associated with anabolic processes, meaning you're building things up. We end up getting water as the byproduct. You do need to know the difference between catabolism and anabolism. Catabolic processes are breaking things down. When we eat food, we go through a catabolic process to break that down. anabolism, so anabolic reactions, think about anabolic steroids. That's what buff people take to get more buff. So anabolic builds up, catabolic breaks down. Okay, don't worry about that. Know the very basics of water, but the most important parts that you need to know about this is going to be the hydrophilic and hydrophobic aspects, and pH. pH is the big one here. That's the money one, okay? So, excuse me. So, what you need to know is the basics of pH, right? And when we're at a neutral pH, that's when we've got a value of 7, and that means you've got an equal amount of your conjugate acid and conjugate base, meaning your protonated hydrogens and your hydroxyl groups. So when something's acidic, it means you've got more protons. When something's low, it means you got more and you got low amounts of base. When it's basic or alcohol, it shoots the scale up. Remember, it's a negative logarithm, so basically it's the inverse of whatever you think it's going to be. So low basic means that you've got a high amount of hydroxyl in there as well. So there's really the pH range of human blood, as it mentions here, is around 7.35 to 7.45. If we go 1 unit low or lower or higher, if that drops to 7.25 or rises to 7.55, then we basically die. So we have very, very strict buffering mechanisms. We have around 3 of them in our body. The most popular one is the phosphate buffering system as well. Basically, and that's because we're constantly breathing in and exhaling, right? So we're always introducing gas, and we need to find ways to be able to maintain our blood pH, and it does that through a number of mechanisms, which we'll go on later on in the semester. We do touch on it when we talk about blood. It's really, really cool, actually. Anyone who wants to talk about it, I'm more than happy to go through it, but you don't need to know it for this lecture. Just appreciate the basics of pH here as well. As we mentioned here as well, it's an inverse relationship with the amount of hydrogen that you've got. So more hydrogen just think of hydrogens as being acidic, and then your hydroxyl groups as being negative. As being basic. As you can see here, we keep our urine based around that, roughly around here, around that 6 on average, around 6 pH. As you can see here, pure water, when it's not acidic or basic, is around 7, and then as we shoot down the scale here, you can see beer, vinegar, wine, pickles are around pH 3. Coca-Cola is around. I don't want to get sued by Coca-Cola beverage company, so I'll just say cola is around 2. So that's why you can clean your floors with it as well if you want. Probably not recommended because it's got a lot of sugar and it's going to be quite sticky, but you can actually clean things with Coca-Cola, as you probably know anyway. It's a very, very good solvent because it actually is quite acidic. Okay, stomach acid is around 1 pH, and then 1 molar of hydrochloric acid is at 0. Okay, ATP, you do need to know the very basics of this. All you need to know is it's basically adenosine monophosphate is adenosine with 1 phosphate group, diphosphate is 2, and triphosphate is 3. Every time that we actually want to power something in a cell, we're basically using ATP. That's our cellular currency. What we do is every time one of these phosphate groups breaks off, energy gets released in the form of heat, and that drives different processes as well. So this is essentially what we end up doing when we're generating when we're utilizing fuels such as glucose. We're generating ATP so that we can keep powering things on. All right, let's go back. So that's any questions on anything on that first part of the lecture? No? All right, just loading up the second part. Yeah, the heat capacity term. So you don't need to know a lot about that, but basically when ATP gets released, it's like a little explosion, and we use that heat as energy. So heat is energy, right? And that's what we use to power a lot of processes as well, to drive a lot of processes. So that's basically what happens when you get Senk. Yeah, you got a question? And Nimajot, did that cover your question? Did it about heat, or were you referring to water? I'm just wondering if you're referring to what I was saying about ATP, or if you're asking more about water, because water obviously protects us from heat essentially. What was the first part of the presentation of? Did you just join in? Did you, Senk? So we're just going. So we're basically going through the lectures, but mostly with the main concepts of some of them. So we're not covering every slide. So if you've covered Senk, if you've covered the lectures so far, then you're on track. It's mostly about the main points that I want you to be able to take home. So yes, Sarah, good question. It's anatomy and physiology is connected for this. So this is really physiology. You got to remember that physiology is basically the physics of living organisms in this case here. So this is really a very, very basic understanding of how you could be able to understand how those physiological processes actually happen in anatomical systems. A good question. It's a bit of a chicken or an egg thing, isn't it? Okay, Nimajit says about the concept that we don't cool fast or heat up. Oh, yeah. Okay, so water basically essentially is, like it fills up a lot of us. Our plasma, makes up, our blood. It's largely water as well. So water basically allows us to be able to regulate that temperature, right? As we were talking about last week as well, the way that we actually maintain homeostasis on body temperature, for example, is via the way that we traffic blood towards our skin. So when we get hot, we actually we want to bring the actual blood more to our skin, which is why you get flushed when you're hot. And that's because our skin has the ability to be able to let that blood cool down. So we sweat, that allows the wind to hit us, it cools us down, but that just doesn't cool us down. It's cooling the blood that's on the surface of our skin. And then once that's cooled down, we can allow that to come internally a little bit more as well. The opposite of when we're hot or when we're cold, what we want to do is we don't want that to be exposed on the skin. That's why you get those goosebumps that form. It restricts the actual blood flow to the skin, limits the actual surface area, and then you start shivering and stuff like that to be able to generate heat. All of that is about warming up the liquid in your body, which will therefore allow your organs and stuff to be able to maintain that normal body temperature. Does that answer your question, does it? It's all because of water. I hope that answers your question, because we better move on. Okay. Fantastico. Okay, let's move on to the cellular level. 2 hours, Sarah. That's 2 hours the exam. So, all right, let's restrict our questions just to lecture content at the moment. Otherwise, we'll get bogged down with other things. Feel free to email me. I don't get on the discussion board as much as I should. You got to remember that there's a lot going on this semester as well. Try and use email me questions you've got. I will jump on the discussion board once a week or so, but if I'm on there every day, I can get bogged down with a lot of stuff. So use it more to communicate with your peers and discuss things as well. Use me for your go-to for questions about lectures and stuff like that. Okay, so what do you need to know? You need to know from this the basic fluid mosaic model of membranes. That's the main thing. And the other thing that you got to remember is cellular diffusion and osmosis. And you're going to have to remember how things get in and out of the cell. So let's skip ahead. You don't need to know every one of these organelles, but you should know. I'll quickly show you. You should know. You should be able to look at this and know that this is the phospholipid bilayer. You got little elements of cholesterol embedded throughout that. An odd fact, you don't need to know it for the exam, but there's an organelle sort of anatomy of the cell. So a good question, Caitlin. An organelle, if we go back, cellular organelles are, they're basically like, think about the way we've got our heart, we've got our lungs, we've got our liver. A cell has its own little organelles as well. The Golgi apparatus is an organelle. The ribosomes are an organelle. The mitochondria are an organelle. Some are membrane bound, some are non-membrane bound. Ribosomes are free floating and knocking about in cytoplasm, whereas your Golgi apparatus, your endoplasmic reticulum, your mitochondria, they've all got their own membrane, and your nucleus obviously as well. Does that answer your question? I hope it does. Fantastic. All right, fun fact, cholesterol actually increases the melting temperature of the phospholipid bilayer, and it also provides more stability and rigidity. So let's call it fluid mosaic model, because it is a mosaic of things, isn't it? And also it changes constantly as well. Every time something wants to leave a cell or enter a cell, it goes through that process called budding. You don't need to know that for the exam. It's just a cool bit of cellular biology. Remember, we've got a whole bunch of proteins here that want to get out. They go and get packaged in a little bubble, like almost bubble wrapped with phospholipids, and they leave it like a little lipid droplet that's full of this proteinaceous cargo. And then they go off, and when they want to enter another cell, they fuse into the membrane, become part of the membrane, and dump their cargo in. Pretty cool stuff, huh? All right. So we will talk about channels, but the things you need to know are what the membrane organelles are and what the membrane organelles are. You definitely will need to know that. Here we go. Here's your membrane organelles. We don't go through all of them here. This is a good example of one. See, the Golgi apparatus has a membrane. This is where a lot of proteins get glycosylated. Pretty cool stuff. Glycosylated means having a sugar added to it. Okay. So your nucleus is another good example as well of a membrane-bound organelle. So let's go forward now. This is the real money part of this lecture, okay? So, excuse me, this getting over this cold, and it is a nasty one as well. Anyone else get sick over the long weekend, just out of curiosity, or was I the only one who didn't get to use the long weekend like I should have? I had big plans too, big plans. Yeah, no one else got sick over the long weekend? You did, Sarah? Yeah, definitely feeling your pain there. It's Manaz didn't get sick. No, Georgia didn't. So, yeah, you guys got lucky. You guys got to enjoy the sun. Nothing worse than having a head cold. Oh, Shay did as well. So, and Fion got their knee destroyed. Jeez, that's not good at all. So, yeah, look, I was bedridden with man flu. It's a terrible thing. It does affect men worse. That's not scientifically proven. I'm just going to state that it is. Okay. So, the things you need to you thug it out. That's pretty funny. Okay, I'm going to shut the chat group now because I'll get distracted. Now, fusion and osmosis is the money part of this lecture, and you need to be able to appreciate the difference between the two of them. So many students, they balls this up every year, and I don't want you to do that, okay? Remember the difference. Diffusion is the movement of particles from a higher to lower concentration, whereas osmosis is the similar thing but across a membrane. So people get a little bit confused about that. And one of the ways that we get things in and out of a cell is largely through diffusion, but we have to do osmosis to get it through the membrane, right? Everything in physiology, things will move under the laws of physics. Things will always go via a concentration gradient. In this case here, we have a very high concentration and a low concentration of sugar in here. No sugar at all. We drop the sugar cube in, it is going to want to go to via a flow to a concentration gradient. That means that it wants to permeate throughout the actual water and be able to have an equal distribution here as well. Remember, everything that you have in physiology, if you have a high concentration, and you put some and then you've got an environment where there's a low concentration, you will get diffusion occurring, which will distribute that so it's all evenly distributed. So that's not osmosis. Diffusion going across the plasma membrane is essentially osmosis. However, the diffusion itself will go from a high concentration to a low concentration. That doesn't just happen with liquid or sugar or anything like that. It also happens with charged elements as well. If you've got a whole bunch of positives over here and hardly any positives here, they're going to basically want to go pass in through the membrane if they can. If they get through, they're going to want to be evenly distributed. So osmosis, as we were saying, is the diffusion of water across a membrane. So think about it that way. Diffusion is essentially the concentration gradient, the flow of things from a high concentration to a low concentration. Osmosis is the same thing, but it involves a membrane. How about that, huh? So if diffusion goes through agar gel, will it spread and take up the whole surface area? If it can, if it allows, yeah. So definitely, if the permeability of that gel will allow it to, then yeah, it will. Okay. So a couple of things that you need to remember. This is really cool for learning about blood cells as well. So basically, there's a number of things that we've got here. Now, this comes down to salt concentration as well. Our cells like to have a nice balance of so basically your ions and your water. When we have that, so you can see here, here's our solute molecules, and our water molecules are orange. When we have a perfect balance, the cell is isotonic. That's why all those sports drinks that are essentially just salty water have like use the term isotonic beverage and stuff like that. That just means that it's going to allow your body to be able to have the correct ionic balance mixed with water as well, which will make your red blood cells pretty damn spanky like this one here. Now, when it's hypotonic, that means you have a reduction in solution, but you've got a lot of water. A lot of water and not a lot of ions will make your cells swell up. Remember, that's because you've got more water over here, and then you've got water entering here. Now, in hypertonic is the opposite. It's when you're dehydrated, you've actually got a lot of ions, but you've got not enough water. What ends up happening is the cell ends up shriveling up as well. Does anyone know, just out of an odd fact, the thing I like about anatomy and physiology is there's always something that people, and I guarantee you probably get asked by family and friends, how does this work or how does that work? This is one of those odd little facts. Do you know that water intoxication is a thing? Did anyone know that? Yeah, so you can die if you drink too much water, right? It's not just your kidney, and it's not just your kidneys that pop. So it's also your cell. This is essentially what happens to your cells, right? When you have too much liquid, and there was actually a case in America where they did a thing called Hold Your Wee for a Wee. A radio station did it where they put these people up on a stage, and it was almost like those eating competitions, but it was a water drinking competition. So they had to drink a hell of a lot of water, and it was in the it was the UK, was it, James? And then what they did was they basically, the people who held their pee for the longest period of time, they'd be able to, without going to the toilet after drinking 5 liters of water or something, they would be able to win a Wii, Nintendo Wii. So that was the Hold Your Wii for a Wii. And a few of the people got really, really sick. One of the people died. And that's because what happens is all your cells will start to swell up. Now, it's not too bad for a lot of our organs that can expand, but what about our brain? Our brain is stuck in our skull, right? There's very little room for movement. And this is actually why fever is catastrophic as well, because when things swell up too much, your skull basically doesn't expand much. So basically what ends up happening for this poor person who died from water intoxication is they ended up getting a brain hemorrhage because essentially their brain swelled up and got crushed by their own skull. So pretty nasty stuff. And yeah, it definitely is unethical. Yeah. So someone asked a question, how much water is too much? That I can't remember off the top of my head. I think if you ingested around 5 liters or something very rapidly, then you're going to be in a lot of trouble. So, and actually, that's where a lot of people as well have died from MDMA, the party drug, ecstasy, that a lot of people do take at music festivals and stuff. A lot of people who drink too much liquid on that end up dying of this as well, and that's because that particular drug slows the production of your antidiuretic hormone, so your body retains a lot of water. That's why if you're on that drug, you don't urinate very much as well. So a lot of the people who have died of MDMA is largely because of water intoxication, even though they haven't drank a lot. Okay, you do need to know how you can link diffusion and osmosis towards understanding how carrier-mediated transport can occur as what we call the facilitated diffusion. Basically, you've got here a molecule will bind, and then it will basically be released. This is through a channel protein. Now, normally, if this is going towards a concentration gradient, it will just flow as normal. However, if it's going against the concentration gradient, that's when we need to use an active process. This requires ATP, our cellular currency, and we utilize ion exchange pumps. In this case here, the ion exchange pump is almost like a piston, right? So you have three sodium positively charged sodiums go out, 2 positively charged potassiums come in. This utilizes, again, there's an unequal exchange here of ions and charge. So because of that, we need to use ATP. Now, why do we want to do this? Why do we want to have this pump going? Well, it's because that's one of the ways that we get glucose into a cell. Glucose comes in, basically sodium binds, that makes a conformational change in the protein that lets glucose bind. Then we utilize this pump, and this pump allows this particular process to be powered. So pretty cool, huh? You don't need to know that in depth, okay? All you need to know is the difference between active and passive transport, okay? You need to know active. Just remember, active means it requires ATP, okay? Any questions on this part of the lecture before we move on to the third part? No questions? Is everyone enjoying this format a little bit more rather than going through every slide and just touching on things? Is everyone preferring this format? Yeah, great. So what I might even do next week or so is I might start to what I might do is cut back a certain amount of slides as well, just for this online session, so that we really just present, like maybe 5 to 10 slides per lecture, which means if we've got 3 different smaller lectures, we should end up having about 15, 20 slides that we can just really rip into. Okay, if everyone's happy with that, we might go forward with that. All right. So passive transport, no, it doesn't. No, that's right, Akira. Passive transport does not use ATP. Okay, let's go back to the content. All right, let's get on to the third part here as well. Which is dominant in us, active or passive? Oh, that's a good question. I couldn't answer that off without making it up. I would say passive, because we're always got things going from a low to going along a concentration gradient. I would probably say I would bet my money on it that passive would probably dominate. When you said channel proteins, is that the same as a carrier protein? Yeah, a carrier protein in this case, this is a carrier here. Whereas I'll just go back. Really good question. If we go back to this model here, this can just be a channel as well. It'll allow things to go from a high concentration to a low concentration. Whereas a carrier is something that will actually bind to it and then move it through. Very, very similar. But yeah, you don't need to know that for the exam or anything. But very good question. All right. Now we get to the good stuff. You're very welcome. So now we get to the good stuff. We're up to the cellular level. And this really is where stuff starts to get really into like more about what you're going to be assessed on really as in terms of getting to that more anatomical level as well. Remember, we start from the bottom up. So, and we've done a lot of that today. Okay, so what do you need to know? Like list the functions of plasma membrane and structural features. Oh, wait, hang on a second. Yep, no, that's okay. Hang on a second. I've got to go upload the tissue one. I clicked on the wrong one. Sorry, guys. This man flu is getting to me. Okay, we've done chemical. We need tissue. That's what we need. All right, here we go. Can everyone see that? Yeah, fantastico. All right, I can tell you now, not even all the caffeine in this coffee can compare to the intensity that you're going to experience with this lecture, okay? So, you do need to know the four major types of tissue in the body. You need to know the functions and the types and functions of epithelial tissue. You do need to understand the relationship between structure and function of each type, but not in a great sense as well. Just know the basics of the four different types, right? You need to know the functions of connective tissue, and you need to know the difference between your connective tissue proper and your fluid connective tissue, which is just basically blood and lymph. Know the basics of this stuff. You don't need to know it in great depth. I'll point out the things you do need to know. Describe our cartilage and bone function, function as supporting of connective tissues. You need to know that, but this will be enhanced when we go around, when we talk about bones and joints next week. Describe the three types of muscle tissue and special structural features and type. Yes, you do need to know that, and the basic structure of nervous tissue. So really, there's a lot to take from this part of the lecture. But I think one of the main things, if you can use this slide here as print this out and put it up on your wall at home, right? Frame it if you want. It's worthy of framing. It certainly is worthy of framing, right? Put it up on your wall and try and remember this stuff. You need to know this stuff for the exam, okay? This has largely a lot of what you do need to know in regards to the things that you should be using as your own study guide. But let's get into epithelial tissue. So epithelial tissue basically makes up a lot of what's going on in our with our skin and all that sort of stuff as well. Now, it's not just our skin. So obviously our epidermis is our skin. However, we have epithelial layers on every sort of every organ that actually has a surface lining, right? So our liver, for example, has epithelial tissue around it. Our kidneys do as well. So all that sort of stuff. Basically, it provides protection, permeability, sensation, and specialized secretions. Secretions means anything that something leaks out. Mostly sweat is the main thing that we've got that we're going to we secrete. We also secrete sebum and a whole bunch of other things as well, but that's for another. That's different. Don't worry about that too much. Okay, as you can see here, you should be able to look at this and go based on the last part of the lecture and go, hey, there's a nucleus. Hey, there's a Golgi apparatus. Hey, here's some mitochondria. Now, this is really, we're looking at two cells that make up part of the skin in this particular case here as well. What you will notice is there's a basement membrane, there's the basal surface, which it attaches to, and then you've got your cell, and then you've got your microvilli and cilia at the top. Now, these cells are connected together in a very, very tightly regulated structure. Now, they're proteins called connexins. You don't need to know those. What they do is they create gap junctions. So that means that if you've ever looked at a brick wall and there's mortar that actually holds the bricks together, that stops them. If you can't just stack the bricks and glue them together and stack them one on top of another, they'll basically fall over. They'll kind of crumble into each other, right? So think about what's going on here is like mortar, right? And your gap junctions basically allow that to happen. What they do is they keep everything close, but not too close. And we have three different forms. There's gap junctions, tight junctions, and desmosomes. I'll just go and show you here. This is so you can see how it all works. Here we go. Here's this is the exact same image we were looking at before. Here's the cilia, the microvilli up here, but down here, as you can see, we've got our gap junctions here. Your gap junctions are holding the cells together. Your spot desmosomes are basically maintaining a good amount of gap between there, so that you've actually, so that they're not just going to squash into each other. And then our hemidesmosomes are what are helping attach the actual the basal membrane to the basal surface. And that's what you can see here as well. This is how your tissue attaches. It actually attaches through to the basement membrane. Excuse me. And we refer to those two parts of the actual basement membrane as the basal lamina and the reticular lamina. You don't need to know too much about that as well. You do need to know that the epithelial tissue will, where the basement membrane will end up attaching to the basement surface. Now, you do need to know this stuff here as well. You need to know the shape and the layers of epithelial tissue. These are quite self-explanatory, so you should be able to remember them quite easy for an examinational context. Excuse me. Squamous means that, like, think of squash. It means they're thin and flat. Cuboidal, square and square-shaped, and columnar, which is those like the example we showed before, tall, thin sort of columns. Now, based on we can have this as either a simple epithelium, which is a single layer of cells, or stratified, which means it's layered. Now, I'll let you go through that on your own time. You only need to know the basics of that. So connective tissues, you need to know about this as well. So do the different classifications have different functions? Yeah, they do, Akira, and we'll go through that a little when we go through the chapter on skin, where we will cover everything about that. I think that's in week 3. I'm pretty sure it's in week 3, or week 4, sorry. We'll go through everything about that as well. So don't worry too much about that for now. You're welcome. So connective tissue is, you've got to know the difference between connective tissue proper, fluid connective tissue, and supportive connective tissue. What does that mean? So, well, you've also got your loose and your dense connective tissue. Know the very basics about that as well. I'm not going to quiz you too much on that as well. Connective tissue proper, this is basically what it is, right? If you have a look at this particular image, you get a really good understanding of what it is, and that is the sum of all these different things. It's got fiber, it's got fluid in the form of ground substance, and it's got all your different cell populations. So good. Who had that question? Let me go. That was a good one. So Sarah says, is this the only time we go through this content, or do we recap in class? So much info. It's the only time we'll go through it in this broad sense, and then we're going to narrow down. So we will cover, so when we go talk about the heart, the cardiovascular system, we will cover your cardiac tissue associated with that. So we will go into this stuff in more detail. So yeah, definitely. Now you need to know the difference between, but the things you need to know from this, though, Sarah, is you need to know some of this real basic stuff in terms of definition. And it is a bit of learning by rote, I know, but you've got to know the difference between loose and dense connective tissue. And you've got to know that fluid connective tissues as well. A lot of people don't quite realize that blood is a tissue. Blood is actually a tissue. Fat is a tissue. Lymph is a tissue as well. These are basically fluid connective tissues. Fat isn't, obviously, but fluid connective tissues are largely your red blood cells, your white blood cells, and your platelets. If you remember platelets, they contribute to blood clotting, like we talked about last week. Supporting connective tissues are cartilage and bone, and cartilage is basically largely comprised of chondroitin sulfate, which is why a lot of those things that don't have a lot of evidence about them, behind them, to try and support cartilage function. Basically, glucosamine fueled with chondroitin sulfate. They try and claim that you will regenerate your cartilage, which you can't regenerate your cartilage. You might be able to provide some lubrication to it, which can decrease the pain, but it will not actually, cannot, without stem cell supplementation, you can't regenerate cartilage. So if anyone knows a way, and if you found a way, then let me know, because I need some. Like, years and years of sport have sort of ruined my joints. Okay, so bone is another guy is considered as our supportive connective tissue. These are quite easy to remember, and don't worry about too much about this stuff. We will go into bone next week, actually, when we get into bones and joints and stuff like that as well. So don't worry too much about this right now. The main things that I want you to be able to take away from this stuff today is those basic definitions of everything, right? So the differences between the different classes and subclasses of everything. So muscle tissue, again, this is one thing you need to remember. Muscle tissue can be broken down to skeletal muscle, cardiac muscle, stem cells have totipotency. Yeah, that's right. Yeah, they do. So, and that basically allows them to be able to do all the wonderful things that they do in regards to differentiating into different types of things as well. So, and all of that really originates with our bone marrow, which is really, really cool. Okay, skeletal muscle, cardiac muscle, smooth muscle. You need to know the differences between this. So skeletal muscle, cardiac muscle and smooth muscle all have slightly different looks and functions, right? So I'll tell you about this now. You do need to remember this. So write this stuff down on a sheet or pin it to your wall, right? You don't need to, what you need to remember is that muscle fibers, skeletal muscle fibers, do not divide, and they're striated. Striated means they've got that particular structure, right? So smooth muscle cells, though, cardiac muscle, though, basically, we'll go back here. So cardiac muscle basically is striated. It's involuntary, whereas smooth muscle tissue is non-striated and involuntary as well. That's a little bit different from what we get in. So hang on, I'm just going to try and rephrase that for you, so it makes it easy for you guys. Your heart always beats, right? So it's involuntary. You don't have to tell your heart to beat. Your smooth muscle tissue is going to be associated with a lot of things like digestion and all that sort of stuff as well. You don't need to tell your intestines to contract and to move and for peristalsis to occur. Okay, striated, yeah, it's that particular look that you've got here as well. So where it's like a long, thin, linear sort of cells. Does that make sense? So yeah, that's what we said there, James. Is that what striated? Yeah. Oh yeah, my pronunciation. Yeah. I get that. So yeah, I mean, that's okay. Sorry about that. I thought you were actually understand. You didn't get the definition. That's okay, but you got that. Okay, so your cardiac muscle is the cells are much shorter, and basically they're always going to be beating. Your smooth muscle tissue, as you can see here as well, looks a little bit different. They're shorter. They've got this spindle shape, and they're non-striated. So in my rogue R, whenever I pronounce striated. Apologies about that, James. Okay. Nervous tissue is, you got to know the basics of this as well. Again, just the basics of it. You need to know the definition, the difference between neurons and neuroglia. Neuroglia is basically the supporting cells. They're part of the nervous system, but they're not nervous. So they're not nerve cells themselves. Definitely know the difference between that. all you need to know about this for now is these three major things: the cell body, the dendrites, and the axons. Now, we have a look at this in this particular image here. Here we've got the cell body, the nucleus there as well. Nervous tissue never, nerve cells never divide though. They don't regenerate. They basically just stay. They'll fix themselves, but they stay put for their entire life, right? So you've only got, you don't split. They don't split and divide. So they just keep going on. All right, they're like a truck that's always rolling. And that's for anyway. What you have here is you got your cell body. Your dendrites over here are going to be connected. Now you got to remember you've got one of these, and then you got another one of them over here as well. Sorry, I put that on cursor on. So one of these is going to actually be in a train of different nerves as well. They pass the impulse along. What happens over here is the axon is going to spread out, and it's going to make contact with either cells to innervate muscle tissue and stuff like that as well, or it's going to actually interact with dendrites of a neighboring cell, a neighboring nerve cell, to be able to pass that information on as well. That's pretty much the basics of that you need to know. We will cover a lot more of the nervous system in depth. If you are feeling at all flustered with the amount of information that you've received in these particular lectures, don't worry too much. Remember those things that I was telling you about, that I said you definitely should know that. Just focus on those, and it's really about the definitions and the differences between different classes of either your tissue, cells, or chemicals, and pH and stuff like that. Know just the basics, right? And you should be fine for this week. Are there any questions at all? Yeah, the quizzes are open book. So just good question, Akira. So quizzes, everyone. So the first quiz is available. It does time out. You get one show only. The moment you open it, you've got to go. You've got 15 minutes, right? Yeah, they're all multi-choice. So pre-class lesson and lesson recording is enough. What I would say with the book reading, yeah, you definitely need to go through the book readings. But the thing that you do need to use for with that is, remember, you're quizzed on the lecture content. So the learning objectives. Yeah, don't worry, Anki. We'll go through that when we go through the system later on this semester. So what I would recommend you use the book reading for to enhance your understanding of the learning objectives. So just so you know, guys, everything. When are the quiz due? So Imogen, go and have a look on the announcement last night, because it's got it all on there. In short, you've got 1 week to get it done. Always study towards the learning outcomes. This is what we base the questions on in the exam. So anything that you find in the actual textbook that's not in the learning outcomes, you don't need to know that. Yeah, so if you've had problems with opening it up and stuff like that, and you didn't realize it was timed, it would time out, then send me an email. Yeah, but you got 15 minutes for the quiz. Yeah, send me an email, Sarah, and Hannah as well. All right, guys, I got to go. I've got to run this same exact lecture while I'm a sniffly mess as well. 10:30. So, any questions? Let me know. I'm assuming I'm going to be better. Tomorrow I will be there to give an introduction at the anatomy lab for those of you who have your lab tomorrow. All right, fantastic, everyone. Have a great day, and I'll see you in the next one. You're welcome, everyone. See ya.
step